Okay, we will do vector biology. This is, in my opinion, it is the a very important chapter, and I would expect lots of questions from here. But I don't know. You cannot say. So, we understand. Please pardon some of the spellings over here. So they are carriers of or vehicles of DNA molecules which carry your gene of interest, and when combined with a gene of interest, a recombinant DNA molecule is obtained, and they take the particular plasmid or a particular DNA into a cell. So it's a vector or a carrier or a vehicle. So the aim of using vector in recombinant RDT means recombinant DNA technology is to obtain multiple copies of the gene of interest. So you have cloned your gene of interest in your particular plasmid vector, and you have transformed them in a bacteria. The result is that in this bacteria, the more the bacteria divides, the more your plasmids containing your gene of interest is replicated. So you will have multiple copies of the gene of interest. So that this is the case of cloning vectors to obtain the product of gene of interest. This is also another thing which I have explained, that what you do is that some of them, if you add a promoter in front of your gene of interest, you can express this particular, the plasmid is translated inside your, transcribed and translated inside your uh, the transformed organism, and you will get lots of your proteins. So in the case if insulin has been cloned in your vector, you get lots of insulin. Then you purify that insulin and sell it in the market. So you also know that what is the characteristic of ideal vector? It should be able to replicate autonomously. खुद ब खुद it can replicate अंदर में. It is not dependent on anybody else. It needs the host machinery, but no other sequence will control it. It should be easy to isolate and purify. You have to be able to isolate. For example, isolating chromosomal DNA from bacteria, it is very difficult. But plasmid, it's very easy. Break open the cell and it will come up, and you do a centrifugation, it will come up in the supernatant. There you can precipitate it with sodium acetate and 2.5 volumes of ethanol, and all your DNA will fall down. Also, along with it, lots of RNA will come. You add some RNAs and that RNA will go away. So, ideal vector, it should be, you should be able to transform very easily. It should have suitable marker like an antibiotic resistance, so that only those genes, only those as cells which are being transformed with a particular. Just hold on. হ্যাঁ বলো. হ্যাঁ হ্যাঁ, ওটা সিডস, এগ প্লান্ট না, ওটা তার সিডস. না না, আমি সরিয়ে রেখেছি ওটা. ওটা এইখানে এখানে রেখেছি, ওটা হারিয়ে ফেলিনি কিনা জানি না, কিন্তু আমি ওটা আলাদা করে রেখেছিলাম ওখানে. হ্যাঁ 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 হ্যাঁ, ওই ওই ওষুধের বাক্সর তলায় রয়েছে এগ প্লান্টের সিডসটা. ওটা ওই সানডেতে সয়েলটা আসলে আমি পুঁতে দেবো. না 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 না, কালকে সয়েলটা আসবে, সয়েলটা আসলেই আরো কিছু পর্তুলাকা সিডস আসবে, সেগুলো আমি ইয়ে করে দেবো. সানডে আমি আর অনিকা করে দেবো. হ্যাঁ হ্যাঁ, এসেছে এসেছে. হ্যাঁ হ্যাঁ হ্যাঁ, ঠিক আছে, একটু পরে করছি, আমি একটা ক্লাস নিয়ে আমি তোমায় কল করছি. হ্যাঁ হ্যাঁ হ্যাঁ, ঠিক আছে. Okay, so it should have a suitable marker genes that allow easy detection. আমাকে শুনতে পাচ্ছ তো তোমরা?
Yes, sir.
ওকে, ওকে। So and that is an antibiotic resistance. And so if you grow these cells in a media containing that particular antibiotic, then you will see that only the cells which have been transformed with this vector containing your suitable marker genes, they will survive. The others will die because they will not be able to grow in that antibiotic media. Only the transformed cells. So you can select only cells which have been transformed, and the ones which have not been transformed are will die. Then it should have the ability to integrate itself either or the DNA insert it carries into the genome of host cells. This is in a later case that you can integrate it into the genome. It should contain unit target sites. So it should contain restriction enzyme sites. Right? And say, for example, this particular host, it should be recombination negative, because moment you transform plasmid, the plasmid then recombines with the host chromosomal DNA, then uh, all cases jaundice, because you want to keep the plasmid separate as such. So it should also contain promoter, operator, ribosome binding site in the case you want to express your gene of interest. So you must have a promoter, operator, ribosome binding site, because ribosome binding sites are needed for binding of ribosomes for translation into proteins. ঠিক আছে? Clear? রুচি? Clear
Yes,
hai?
sir. Yes,
Okay.
sir.
Okay. So, general construction of vectors, which I have already talked in the college, you should have an origin of replication. We have seen origin ori sequence. You should have selectable markers. So now vectors can be on the basis of our aim with gene of interest. You can classify vectors into cloning vectors and expression vectors. Cloning vectors only your gene of express is cloned inside it, a recombinant DNA technology is made and it is transformed. Its only function is that you break open the cells and you get more number of copies of this particular plasmid. Ek plasmid say you can get multiple number of plasmids. Then you have expression vectors. That the function of expression vector is to say the gene of interest has to be expressed inside the bacteria. So now you can classify vectors on the basis of host cell use. So vectors for bacteria, vectors for yeast, vectors for animals, vectors for plants. So you can transform bacteria, you can transform yeast, you can transform animals, and you can transform plants. And on the basis of the cellular nature of vectors of host cell, you have prokaryotic vectors and eukaryotic vectors. Bacteria mostly we use prokaryotic vectors, but human genome sequences and all these things, they have been used by from it eukaryotic vectors. They have been yeast artificial chromosomes, mammalian artificial chromosomes, they use them. So prokaryotic vectors are plasmid and bacteriophage vectors. So they have been used. Most commonly, they are small, circular, double-stranded DNA molecules. They lack protein coat existing naturally in the cytoplasm of many strains. So they are small, circular, double-stranded. We have seen about plasmids. So most important are PBR322 and PUC vector. So let's have a look at PBR322. This was the first widely used plasmid vector. PBR322 has a relatively small size. This is 4.363 base pairs. This also has a reasonably high copy number, that is 15 copies per cell. Quite high. One cell will give you 15 plasmids. 10 cells, 150 plasmids. 1000 cells, 1500 plasmids. And you'll have into the 10 to the power 8 cells, 15 into 10 to the power 8 number of plasmids. That's quite an appreciable amount. So P is the plasmid, and the BR is from Bolivar and Rodriguez, the two researchers who developed it. And 322 is the number, plasmid number which was developed. They developed different other plasmids also, but this was 322 number in the laboratory. So if you look at PBR 322, you will see this has got an Cole Ori sequence. This is the Ori sequence. You have a tetracycline sequence over here. Tetracycline antibiotic resistance and ampicillin resistance gene over there. And this EcoR1 hint tree, in this place, you will have your multiple cloning site. ঠিক আছে? Clear? Ruchi?
Yes, sir.
Okay. Okay. And it has got cloning sites. You have seen ampicillin and tetracycline. It is widely used as a cloning vector. It is not an expression vector. Okay. Due to its small size, it is easy. You can purify it easily. It will break less. There is breakage while purification. And two selectable markers. Easily you can select it. That you grow in a media of ampicillin and tetracycline. Only the ones which have been transformed cells, they will grow. Disadvantage, it has very high mobility. There is a limitation in the size of gene of interest and it can accommodate. So if your gene is of interest is very large, you cannot clone it inside. It becomes very bulky. So it has very high mobility. So the moment your the antibiotic goes away, this the plasmid will be lost in the media. Then you have the PUC vectors. They have been obtained by modifying the PBR322 vectors. They are smaller than PBR322, 2.7 kb. It produces 500 to 600 copies, high copy number. You will see that this has got the multiple cloning site over here. It has got the promoter and operator of lac, ampicillin resistance. It has got the lac Z gene, and it has got multiple cloning site. It has got selectable marker as ampicillin resistance, and it has got an Ori sequence, origin of replication. This is the PUC vector. It can be used as a cloning vector and expression vector because you have the lac promoter. If you clone it over here under the control of the lac promoter, you can express this particular protein. Problem is it cannot accommodate larger than 15 kb gene of interest. So small genes like insulin has got 50 amino acid. That is around, that means 150, the DNA will be 150 into 3. So 150 bases. So perfect vector for insulin cloning and insulin expression. But 1500 base 15 kb, that is 1500 base pairs, it won't be able to do it. Like there are cloning like all sodium are membrane protein channels, the genes for the membrane protein channels like sodium voltage channels and all these channels, they are very huge genes. So you cannot clone them here. You need different strategies. Then you have phosmid vectors. This is a phosmid vector. They are similar to cosmic vector. One second, one thing, page is missing. Yeah, uh, plasmid and vectors. They are 40 kb of random genomic DNA. So they are 40 kb of random genomic DNA. They are also known as F plasmid. You can have 40 kb insert inside. Here you cannot have more than 15 kb, but inside these phosmids or cosmids, you can have about 40 kb inserts. Then you have bacteriophage derived vectors. They are commonly known viruses that infect bacteria. Like viruses, phages are very simple in structure and consist merely of DNA. Lambda phage vectors, bacteriophage M13 vectors, some names. Lambda molecule is 49 kb in size. So what you do is, this is the lambda bacteriophage. In the DNA, you remove all things, important things, only keep a few things about important only for infecting a bacteria. So what you do is you simply in this you isolate the DNA, you remove the ones which kill the particular bacteria. What you do there is you remove all those sequence, only those DNA sequence in which you clone your gene of interest. You can clone about 50 kb, that's quite large, and then you infect the bacteria. So what happens is that mm, the bacteriophage which infects the bacteria will die, and in that region there will be a plaque or a lysate. You take that lysate, inside that lysate you will find lots of copies of the lambda phage vector with your gene of interest cloned a lot. But this advantage is you will have lots of advantages is that you can have a huge amount now size of insert. So let me show you a picture of bacteriophage transformation. Acha, are you people able to see it by vector, or you see things? Hello?
No, sir.
তো এতক্ষণ বললে না কেন? You
सर मेरा आज जियो का नेट खत्म हुआ है तो आपको लगा मेरे को दिखा नहीं रहा।
बोला मुझे लग रहा था कि दिखाई कहाँ नहीं दे रहा है। मैं बक 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 करता जा रहा हूँ। ओके, एनीवे I'll start it again once more. Okay. Now we are at, well, let me show you a picture of how bacteriophage vectors work. So इतना जितना मैं अभी तक पढ़ाया हूँ ये सब I have covered it in the college. Okay. Now I will be about things are I'm going to talk about new things. Bacteriophage transformation. So you have seen plasmid. It gets inside chromosomal DNA. This is the normal thing. Now here what you have, the viral DNA, this virus, these packages, you have packaging material. Along with packaging material and DNA, you infect the particular bacteria with this virus. So viral DNA copies, what happens, you will have phage, lots of bacteriophage containing your gene of interest. Bacterial DNA भी आएगा इसमें, इसमें भी आएगा. So bacteria from other cells will go inside. There is homolog. This is conjugation. This is also not a good picture. Not finding a good picture. Are you able to see this picture?
Yes, sir.
So these phage, they will infect these bacteria, and then they will make more phage particles. But inside this phage, you have your gene of interest cloned. So result is So this, you have the origin of replication. You have seen a plasmid map. This is your bacteriophage. Inside this, they have this DNA, where I'm not finding any बकवास है ये सब. Ah, yeah. So phage vector, it's linear. You break it with restriction enzyme, and you put your gene of interest here and infect bacteria. So if when you infect bacteria, this phage DNA, what it will do, it will keep on replicating more inside the bacteria, and then it will lyse the bacteria, and you'll have a lysate. In the lysate, you will have lots of bacteriophage which has got lots of your particular mm, uh, your gene of interest. Okay, we are going to the next one, phagmid vectors. This phagmid vector, one second here, let's do a play slide. So you have the cosmid vectors. So it is similar to it. They basically, they are a plasmid that carries a cos site. This cos site, it contains selectable markers such as ampicillin resistance gene and an origin of replication. Cosmid lacks all the lambda genes, so it doesn't produce plaques. So the lambda bacteriophage, it produces plaques because it has got the lambda genes. As a result of which the cells are lysed, and then you'll get your plasmid. But in cosmid vectors, you cannot. It doesn't have those lambda genes. Rather, in there, in those genes, you put inside your gene of interest. So uses of cosmid vector is, it is used for the construction of genomic library in eukaryotes. An advantage, you can clone up to 40 kb, and you can easily screen because it has got ampicillin resistance. You have phage mid vectors. They are phage from M13 bacteriophage and mid from plasmid. It contains 1500 base pairs. You have got the origin of replication, lacks Z gene, multiple cloning site, origin of replication, and ampicillin resistance gene. They are used as cloning vectors, sequence in vectors, expression vectors. It can be used to provide single or double stranded material without any recloning. They are truly plasmid with phage genes. They are linear. Eukaryotic vectors, which is more important, yeast vectors, yeast artificial chromosomes. You can have yeast cloning vectors, yeast expression vectors, and yeast artificial chromosomes. They are unique target sites or restriction enzyme sites. They have got high copy numbers, and they use also selectable markers. You also have animal vectors like insect baculovectors. You can transform these in insect vectors, in insect cells. You have the bovine papilloma virus vector. You can clone, it's around 79 kb long, and you can clone large amount of sequences. SB40 vector. Then you have, this is a SB. You have to just simply practice these figures. You have plant vectors. You don't need to study them. Artificial chromosomes দরকার নেই. ওই yak and plasmids, they are most important in your syllabus. ওকে, কোয়েশ্চেন্স আছে? এনি কোয়েশ্চেন্স?
नो सर।
বর্ণালী? হ্যাঁ, পিইউসি তে ক্লোন করলে ইট ইজ বোথ অ্যান এক্সপ্রেশন ভেক্টর অ্যাজ ওয়েল অ্যাজ ক্লোনিং ভেক্টর। ইউ ক্যান ক্লোন অ্যাজ ওয়েল অ্যাজ ইন পিইউসি ভেক্টরস। अच्छा, टुमारो फ्राइडे 7:30, ठीक है? वन मोर क्लास।
ओके सर।
सबको थोड़ा बोल देना ग्रुप में। বর্ণালী বলে দিও, সবাইকে থাকতে বলো। হ্যাঁ, কাল কাল ফ্রাইডে 7:30 আমি জিম থেকে এসে আই উইল টেক দ্য ক্লাস। ওকে দেন, বাই বাই। কি বললো? ও সার্টিফিকেট কোর্স গুলি মারো, ওইসব করতে হবে না, ফালতু যত সব ঢপের। তোমাদের অ্যাকচুয়াল পরীক্ষা সামনে এসে গেছে, এখন সার্টিফিকেট কোর্স হচ্ছে, ঠিক আছে? ওসব থাকুক, সার্টিফিকেট কোর্স 7:30 সময় থাকবে না, কিন্তু সার্টিফিকেট কোর্স ওইসব ছাড়ো। এখন মোর ইম্পর্টেন্ট হচ্ছে ও 8 PM এতেই তাহলে করবো, 8 PM করবো। এইসব সার্টিফিকেট কোর্স হচ্ছে, এই নাটক যত সব, এদিকে পরীক্ষার কিছু ক্লাস নিচ্ছে না, সেগুলো আগে করতে হবে, ঠিক আছে? ওকে। ওইসব সার্টিফিকেটের পেলেও কিছু হবে না, না পেলেও কিছু হবে না, কিন্তু এখানে নম্বর খারাপ হলে খুব প্রবলেম হবে।